Hello and welcome everyone. My name is Joanna and today we are going to learn about the role of glycolysis in microbial energy production. First, let us start by understanding what exactly is glycolysis. Glycolysis is the initial stage of cellular respiration where our main component, glucose, is broken down to form pyruvates, thus producing energy in the form of ATP, and this occurs primarily in the cytoplasm of cells. Now, why exactly is this process of glycolysis so important for microbes? In microbes, glycolysis acts as a central pathway for energy production. This is especially important for microbes that survive in environments where oxygen is scarce or unavailable, that is, in anaerobic conditions. Microbes in these oxygen-poor conditions, or microbes that are like mitochondria, like many prokaryotes, rely heavily on glycolysis for production of ATP, that is, energy production. Not only that, even aerobic microbes heavily rely on glycolysis as the first step to produce energy from glucose before utilizing aerobic processes for further ATP production. So, firstly, glucose is phosphorylated by the enzyme hexokinase using one molecule of ATP and converted into glucose 6-phosphate. This phosphorylation traps the glucose within the cell and it makes it more reactive. Then, as you can see in the reaction, glucose 6-phosphate is converted to fructose 6-phosphate, which is then converted to fructose 1,6-biphosphate. Now, this molecule is converted to G3P, which is a 3-carbon molecule. This molecule undergoes further reactions to form pyruvate. In the process of these reactions, ATP, as you can see over here, and NADH is released, which is what provides the cell with energy. Now, the products of glycolysis are firstly ATP. ATP molecules are produced per glucose molecule through substrate level phosphorylation. So, a total of 4 ATPs, as you can see here and here, are generated, but 2 ATPs over here and over here are consumed in the initial stages. So, the consumption of 2 ATPs gives us 4 ATPs in glycolysis. The second product is NADH. 2 NADH molecules are produced which can be used in aerobic respiration or fermentation after glycolysis is done. Then pyruvates, 2 molecules of pyruvate are generated which serve as substrates for further metabolic processes like the TCA cycle or for fermentation. So let us talk about this energy that is produced in the process of glycolysis. First is in substrate level phosphorylation. Glycolysis involves 2 key steps of substrate level phosphorylation, where a phosphate group is transferred directly from an intermediate substrate to ATP to form ATP. In the absence of oxygen, that is for anaerobic organisms, this is one of the main ways for them to produce energy. The total gain of 2 ATP molecules per glucose molecule is crucial for anaerobic organisms that do not rely on the electron transport chain. The second is NADH as an electron carrier. In glycolysis, NADH plus is reduced to NADH during the oxidation of glucose. Now, NADH can be used in aerobic respiration, that is in the electron transport chain, to produce more ATP, or in absence of anaerobic atmosphere, it can be involved in fermentation to regenerate more NADH, continuing the cycle which produces more NADH. Now, the NADH produced is a key link between glycolysis and other energy-producing pathways like the electron transport chain or fermentation, depending on whether oxygen is available or not. Let us now talk about the microbial applications of glycolysis. For aerobic microbes that are living in oxygen-rich environments, after glycolysis, the pyruvate is transported to the mitochondria in eukaryotes and cytoplasm in prokaryotes, and it enters the citric acid cycle, that is the Krebs cycle, for further ATP production. The pyruvate is then fully oxidized to carbon dioxide, and the high electrons are passed through electron transport chain and generating much more ATP. On the other hand, for anaerobic microbes that survive in environments without oxygen, glycolysis is followed by fermentation. The pyruvate undergoes fermentation to regenerate NAD+, and this allows glycolysis to continue by producing more NADH, and thus the cycle continues. Examples of fermentation products in microbes are lactic acid, which is found in lactic acid bacteria and also the muscle cells of humans, ethanol, which is found in yeast and some bacteria, and butyric acid, which is found in some Clostridium species. This fermentation pathway allows energy production to continue despite the lack of oxygen, but it is still less efficient than ATP produced compared to aerobic respiration. Nonetheless, these have industrial relevance. The microbial fermentation processes have been harnessed and made useful for industrial applications. As seen in the previous slide, ethanol, which is found in yeast, is used for brewing and biofuels. Lactic acid is used in dairy and the food industry for yogurt and cheese. Acetone and butanol are used as industrial solvents. In conclusion, glycolysis is essential for microbial survival, both in aerobic and anaerobic conditions, as it provides a way to generate ATP from glucose, that is, produce energy. This pathway helps microbes to adapt to fluctuating environmental conditions, that is, they can switch between aerobic respiration and anaerobic fermentation as and when needed. Beyond basic energy production, glycolysis also helps in biosynthesis and producing valuable metabolites. Understanding glycolysis is crucial for biotechnological applications, especially in fields like biofuels, fermentation, and microbial biotechnology. Thus, we are at the end of our lesson. Hope this video was as educational and fun for you to watch as it was for me to make. Thank you.